precipitation, I mean, we'd expect a decrease in precipitation, we'd expect a decrease in stomatal density and probably an increase in stomatal length, kind of like opposite than what we found.
Yeah, I mean, it could be true. There's, I wouldn't know about stomatal length too much, but I feel like the stomatal density part, that makes sense to me. I guess it also depends on like how the plant is adapted to the environment as well.
Right.
And, just basically the type of plant it is. Because if you see like in more desert kind of environments, the plants have very like reduced stomata to reduce water loss as much as they can. So it'll probably vary from species to species for sure.
That also too, different species are going to have different forms of photosynthesis, where some species, I think it's really cool how some species of plant, they'll open up their stomata at night, but during the day it's closed, and you kind of have like that delayed photosynthesis. So different plants are going to do different things.
Yeah, that's true. So results would vary from different species and also different areas of the US. So I guess for the different areas of the US part, I think, yeah, it would vary as well. Even though we're looking at only Texas, Texas is really big, which is I like how we were able to get such a great range of samples across the precipitation gradient.
Right.
So if we just kind of like extrapolate it, I think it would also reflect that it will vary across the US as well.
For sure. I mean, Texas in some cases is as big as other countries around the world. So
Yeah, the country I used to live in before I came to Texas, Jordan, is like way smaller than Texas.
I think like east to west from Texas is like a crazy amount of time. How long does it take? Something like 15 hours or something to get from one side to the other.
Yeah, exactly. So definitely, I think locations will have a effect on the results that we see.
And you don't have to stay the whole time, Kelly. Thanks for being supportive.
Oh, yeah, for sure. Of course, the longer you stay, the happier we'll be to talk to you about this kind of stuff. But don't feel pressured. No, it's okay. You don't have to apologize. But Alannah, do you want to go ahead and take this next question?
Humidity on the results. Humidity does play a role as far as the stomata goes, and I'm not 100% on little blue stem, but I know on leaves in general, when we're looking at a more humid climate or even humidity throughout the day, if you have more moisture or water within the air, that plant, I know it has to do with the opening and closing of the stomata. So if you have more moisture throughout the air, a plant might be more inclined to have more open stomata because you're not losing as much water. Whereas if you don't have as much water within the air, the plant's going to be more inclined to have a more closed stomata to prevent water loss. Kind of like in the bottom right graph there, you see water within the plant is either being retained or leaving through that open pore. So if the pore is open, it's going to leave. If it's closed, it's going to stay in. If there's more water within the air, it's more inclined to stay open. That makes sense.
Yeah, great answer. I think that covered it pretty well.
Which is also, I was looking at the stomatal length and the width, and which is kind of why I was more thinking about like the open and closed nature of the pore in this particular grass, because I'm thinking if we're looking at like longer stomata, I would think my first reaction would be that the stomata would be more closed. when we're looking at a shorter stomata length, I would be more inclined to think that the stomata is more open. But since in our other graph, the width didn't correlate with the precipitation gradient, it kind of leads me to think, is it leaning more towards in Dr. Tobin's direction, where he was saying that the plant's growth is almost inhibited or stunted or whatnot. The drier the area, or the less precipitation in that specific area. Because it's like, if the stomata length is changing but the width doesn't, I feel like that would be like the stomata are just getting smaller, or like haven't grown as much or formed as much. And then, and I also too, like looking at like the microscope images, whenever I was taking images on the microscope, there was some species, or there was some sites of the plant where the stomata didn't look as fully formed as that. Like almost the whole leaf, the stomata looked really like immature, like very not fully formed at all.
Like way smaller.
Yeah.
Oh, that is interesting. I wonder, like, I wonder if that had anything to do with like taking the impression as well. Because nail polish impressions can be a little hard sometimes.
The impressions, though, they were really good.
Uh-huh.
But it's almost like the stomata itself. I wish I could pull up a picture right now, or I like noted down which plant like was doing
Mhm.
that. But the
Yeah.
stomata itself was like very just immature, like not like it looked itself not fully formed.
Mhm.
There was a lot of impressions that like were not very good though.
Yeah, I know. It's so hard. Like when I went through them myself, my eyeballs would just pop out of my head basically.
Dr. Tobin's like, why is it taking so long? And I'm like, I don't think you've tried to like actually zoom in on these stomata and take like actual pictures of them, because it's like sometimes a whole like leaf, like on one plant, I had to just completely disregard it because I'm like, there's no way I can get an image on it. Everything looked like bubbles and shadows, and it just it was hard.
Mm. Yeah, no problem. Thank you so much for stopping by.
Thanks for stopping by, Kelly.
If you have any more questions, feel free to pop back in later. All right, you too. Bye.
Bye. Where is Jose at? Jose said he was gonna stop in.
Well, seeing as we're here until like 2 o'clock, he
No.
has the time to meander around a little. But what's it called? We are recording this, by the way. I don't know if it give you a notification when I hit record, but I think it usually does. But just letting you know.
Okay.
Okay. Because I just sent out an email saying to do that. But yeah, great first round. Oh, now's a great time to try out
The zooming in and
the
out.
zooming thing, yeah. Oh, it works on my computer.
Oh, that's good.
I'll just pull that up right now. And I think you'll be able to draw on the screen if you want to. Like when you were explaining the lower left image, like the stomata and how you did the lengths and whatnot. I think it would be
Right.
really cool if you like show them exactly like how much you would take from the stomata.
Let's,
Let me just share my screen. Okay, so let's say I put it into presentation mode and then you talk about the stomata at the lower left corner and I zoom in like that.
yeah. I don't think it lets me draw though.
Oh, there's no like annotate option.
Oh, annotate. Yes. Draw.
Oh, okay. Yeah, that looks good.
Okay.
Yeah, and then make sure like keep it in red so that it's easy for them to see.
Let me, do you want me to keep it on there or?
Hi, Dr. Tobin. No, you could
Hi,
take it
Dr.
off
Tobin.
now. But yeah, like I was just saying, like you could just redo it every single time somebody comes in because I just think it would be a nice part to add to the presentation.
Yeah, I think so too.
Mhm.
I think like annotating also kind of like helps me talk too, when because
Yeah,
I'm usually like, oh, this here, that there.
same. I keep on going like this at my screen and then I forget.
I could be like this side of, oh, I'm not gonna annotate. Okay, I gotta
Yeah.
remember to do that.
Mhm.
Dr. Tobin said, nope, I'm heading out.
Yeah. He's had enough. Hmm. Hi.
Hi,
How are you
Oh.
doing?
I think it's working now.
Okay, yeah. Looked like
I
you were having
couldn't-
trouble earlier.
Yeah, I couldn't even unmute myself there.
Uh-huh.
That's the new computer that they just dropped off. So now I'm on the other one. Yeah, so have you been getting people to visit?
Well, we just had 3 people come in
Mhm.
and they left like right before you came in. So now we're just
Okay.
sitting around.
Yeah, it's kind of, yeah, odd because unlike a real, in person conference, you can kind of look down the row and see if anyone's coming and
Yeah. snag
can
somebody here or there.
waylay them, yeah. So, that's a little less possible here.
Mm.
Yeah, so, it looks like this is all being recorded. This is gonna just be,
Yeah, we were told to record this.
okay, I see. So is that's going from your Zoom onto the cloud then?
I just recorded it like onto my computer. That was the option I selected. Is that okay? I've never recorded
I
a
don't
Zoom
know.
meeting before.
Yeah, all right.
I guess
So,
I'll send it if they need it.
so have you had some good questions?
Mhm.
Yeah, okay.
Yeah, some
All right.
questions were like, would different species show the same type of variation, and would we see the same stuff across the whole US? Like, there were some good questions.
Okay. Yeah, those are good questions. I don't know if we would see it across different species. Um, yeah, but that's the next study, right?
Right. Just go
Yeah.
up and onward.
Yeah, we go look and see if we see different or the similar trends in a C3 species versus C4. Yeah, it'd be interesting. All right. Well, that's great. It sounds like it's going well so far,
Mhm.
but you need more visitors. Yeah. And
Yeah.
so I thought the presentation went great. And
Okay.
I like the planted question. That worked out well.
Oh goodness, was it obvious?
Well, when you each asked each other a question, yeah.
Uh-huh.
But I knew something was up. Yeah. But I noticed that there were other planted questions out there. I mean, if I had to guess, like half of them were planted.
Yeah, I honestly thought so too. It was a little bit of a last minute collaboration.
No, but I think that's a great thing, it gives you a way to address another topic in after you've already given your whole presentation, right? It
Yeah.
gets you a way to use that extra time and not have to answer some crazy question,
Uh-huh.
that who knows what's coming. But
Yeah.
so yeah, that's good. It worked out well. I thought Huda did a good job too.
Yeah, I loved her presentation. I thought it was really cool.
Yeah. Yeah. So, well, I guess I'll leave you to it
All right.
to continue interacting, and I'm gonna go check out some other people's posters so that they have some visitors.
Before
Okay.
you leave, I had a quick question about stomatal length.
Yeah.
So for our stomatal length, are we attributing that to like how open or closed the opening is, or is that just the size of the stomata?
That's the size of the stomata.
Okay.
It may change slightly in length as it distorts to open. But a lot of those epidermal cells, they have cell walls. They're pretty tough, especially when they're
Yeah.
fully turgid. So we're the only deformation, only some sort of deformation would cause a change in length. And really we're trying, and when we do those impressions, they're always on the stomata closed. I never do them while they're still open. So we're any change in length caused by opening and closing is not included in our measurements, and they're all measured closed. As you can see when you look at them, in the impressions, right? You've never seen one that like looks open, right?
Right.
And so we're really looking at a change in the morphology of the leaf and in those particular cells that's permanent. And I because they deposit a cell wall, I don't believe that once they've they've fully expanded and then the cell wall has been put in place. After that, I don't think there's a way for them to change in length to any great degree. In other words, the leaf cannot be remodeled after it's been sort of set in place. So so if a plant needs to dramatically alter leaf morphology and anatomy, generally what I've observed is that they'll drop their leaves and grow new ones. And so, because, you know, it's just, they're not able to change very dramatically the way the tissues are built. But again, that's something I would like to, it would be interesting to look and see if anybody has looked to see whether very much change can occur in those stomatal sizes, but I'm not aware of it. Yeah, so we're really focused on different genotypes that occur across this gradient and lead to a different phenotype. It could also be phenotypic plasticity, but it's only plastic in the sense that they can grow new leaves that are different.
Right.
They definitely can respond to light levels, right? But once a leaf is grown in a particular light level, you move the plants to a higher light level and they'll drop those leaves and build new ones. They won't, there's no way for them to remodel the leaf. So that's my current working understanding of it all. Yeah. So actually, I wanted to ask you, Alannah, how have you been progressing with taking the pictures on that new microscope?
Oh, I finished all of that. So all of that's
did?
done.
Oh, great.
Arnold still has to do the measurements on them.
Yeah. But what I'm worried about is we haven't told him that he now needs to change his method.
So I kind of mentioned that to him. He didn't really ask me any questions about it, but which I'm assuming is because he hasn't gone in and done it yet.
Mhm.
But I did mention to him that his scale is gonna have to change, and that we have like a picture of the like the 10 micrometers or that, I don't remember what that's called, the little ruler for the
The micrometer, I
microscope,
think
the
it's called.
micrometer.
Yeah.
So we have a picture of that in there, and that he has to change his scale to do that.
Okay, so you mentioned that. That's great. I was worried that he's gonna suddenly give us like these bizarre lengths with no, having not realized, that situation has changed. And I guess I got some advice from Adriana about how we might improve our image quality, Dr. Visbal.
Mhm.
But if you finished it all already, then it's sort of a moot point. I think the next time we start a big project like this, we're gonna re-examine the microscope that we use anyway. But if you finished them all, that's great. And we don't need
Yeah.
so if what's that?
She came in while I was like doing my measurements, and she said that the ecology students were using a different microscope.
Yeah. Yeah, briefly, we used a microscope in the lab that she's working in.
Oh, okay.
It's these AMscope microscopes, and they have basically it's an iPad pasted on the top.
Oh, okay.
And they're even lower quality instruments than the one the Echo. But the Echo is optimized for cell culture, which is why it had that weird way of where it can rotate, it actually can take pictures from the bottom,
Right.
right? Where you rotate the objective to the bottom and are looking up at a cell culture because the cells will come down. Yeah. So I think that was one reason we were having issues, is because it's really optimized for something else,
Right.
the microscope. But it's gotta be higher quality than those AM scopes. Although the image was not that much, not that great. Yeah. But I think it was good enough is what we concluded, you know?
Good enough for measurements.
Yeah,
Finish off the data set.
exactly. And we're not, we're not relying on being able to, there's a pretty definite edge that we're shooting for, and you either see it or you don't see it.
Right.
And having a slightly better image quality isn't going to mean we misidentify the edges. If we were trying to ID a particular cell type, where you're taking in all the elements of the cell, then yes, image quality would be a big concern, bigger concern. Yeah. All right. So, well, that's good. Well, thanks for banging out those images.
Yeah.
We didn't get Arnold to, that there's that gaping hole now in the poster, right? That we'll be able to analyze it soon and see if we have any different results. We're so close on the first set.
Yeah.
Yeah. I like how you said that, Hatoon, where, you showed there was a trend, but it was just marginally insignificant. Yeah.
Marginally.
Was my
Yeah.
voice good, by the way? Like I tried to put on my very official, making a phone call voice.
Your
I
voice
didn't sound
was-
as dead, right?
Okay. I'll say you weren't as dead,
Uh-huh.
but I think next time you got to sound like when you're doing it, make yourself sound like a crazed clown, right? Then
Okay.
you'll be hitting enough.
That's
how
okay.
that's how, yeah, I have the same issue. I
Mhm.
actually had, when I was first teaching, I don't know if you know Dr. Greg Dement. He actually runs the Center for Teaching and Learning Excellence at UHD.
Okay.
Yeah, but he's actually has a PhD in immunology. He's a full-on
Cool.
science PhD. And then he just, took a different path. I don't think he really enjoyed his PhD that much.
Oh no, that's so
Pretty
sad though.
messy and whatnot. But anyway, he's great and very quiet, very reserved. But when he says something, you have to listen to him carefully. And when you think about it later, you're like, wow, that was really great advice,
Mhm.
but it just doesn't, you don't notice it because he's being so quiet about it. But anyway, he told me, because when I first started teaching at UHD, he was running a program that he wasn't the director of the CTLE. He was actually just part of a program to improve teaching. And he would actually sit in your classroom for the entire time that you were teaching. And then he was like a supplemental instructor on,
Mhm.
And yeah, so he gave me some advice on maybe bringing a little bit more liveliness to my voice. And I had to just, take it much higher than I would naturally.
Yeah. So
Yeah.
basically clown level.
Clown
it.
level, yeah.
Yeah.
Yeah. I don't know what you think I sound like in class, but yeah,
You
I'm
sound,
on.
I think you're great. I never thought clown when I observed your
Okay.
lectures. So you're good. But it's engaging. So that's what matters.
I think
Yeah.
live lectures are really good. I think the recorded lectures are a little monotone.
Mine, yeah,
Yeah.
definitely. Yeah, I totally, I definitely agree with you. They could use some work, but I'll tell you, those are tough. Yeah. So but I'll tell you, the other thing is, if you listen to me at 1.5x,
Actually, all of your lectures, I had to speed up. I put it on like 1.5 or 2x to listen to them because I was like, oh, this is gonna take forever.
yeah, I find that I don't sound too bad at 1.5. Yeah, yeah, which is how I listen to myself when I'm
Mhm.
like checking things. Yeah. All right. Yeah. So, it's one of those things where I give advice, but I realize that, I'm not perfect myself in some of these things that we're talking about, like intonation and liveliness, etc.
I appreciate it nonetheless.
All right. So, but I just did to finish up with the comment though, I did think you had improved, but I think you could take it even farther and
Right.
not even be reaching a level of too much, right?
Mm,
All right.
that's good to
So,
hear.
well, let me get, the other thing is, how do I send people to your poster? I can't catch them in the hallway, as it were.
We can just paste it on my forehead and I'll walk around with the poster.
All
But
right.
we have it on the SharePoint if you want to send it to anybody electronically.
Okay. No, I'm just thinking now, there's nobody showing up. Let me go see what's going on in other places.
I was just
I'll-
texting Huda and she said the same thing. So I think it's just too many poster sessions and not enough people to go
Yeah.
distributed evenly, like to each poster session. Yeah.
All right. Well, I'm gonna go check, just be a presence at some other students' posters. So that,
Okay.
and then, yeah, if I see anyone there, I'll try and send them your way.
Sure. Sounds good.
Right. So
All
now
right. Thank you.
all right. See ya.
Bye.
Bye.
Hold on. I'm going to be right back.
Okay.
Just like 1 minute. Okay. Okay.
I just texted like in the group chat.
Yeah, I saw. Great strategy. I'm also recruiting people. There's a girl in my genetics class. She said she wanted to come and see the poster. So I was like, yeah, come on in. Yeah, come over. Let's see. There's somebody else that I know. She told me she's interested in doing some research next semester and she liked Dr. Tobin. So I'm like trying to get her to come here and see what she can expect basically. So maybe that will work out.
Hello.
Hi, how are you?
Oh,
So do you want us to get into like some sort of presentation, or do you have some questions already in mind?
I guess a kind of presentation. I'm just
Okay.
looking over it.
That's good. So I'll just go ahead and start first. My name is Hatoon Badawi, by the way.
And I'm Alannah Barrera.
Mm. So the main focus of our research was to show that variability of functional traits can be seen in between individuals of the same species and not necessarily among different species, which is usually what research focuses on. And functional traits are clearly defined characteristics of plants that strongly influence their growth, reproduction, or survival. So the species that we focused on for this was little blue stem, and we have a picture of it over here under the introduction paragraph. And the functional trait we focused on was stomata, and we were trying to understand how little blue stem responds morphologically to drought tolerance. So we collected samples across the precipitation gradient, and you can see it in the upper right corner, a map of Texas, which shows a precipitation gradient, and the arrow kind of shows the general direction in which we took samples. We took leaf impressions, which we made using clear nail polish, and we observed those under the microscope at 400x, and we counted stomata, and we also took images of the stomata and used those to measure their length.
Okay, and then we have our results in the center here. So for the first two graphs, we have our late summer data and our spring data for stomatal density, or how many stomata were in a particular area on a leaf for different sites. For the first graph for late summer, we don't see a correlation between precipitation and stomatal density. But for our second graph, we do. So we see that the more precipitation there is, the less stomatal density there is. Or you could look at it as the less precipitation, the more stomatal density. And then the last graph that we have is for stomatal length. Stomatal length, I'm going to kind of annotate here. Stomatal length in this left, there we go. In this left picture here, we're referring to this length right here across the guard cells. And we also had one for width as well, which would be this here, but we just don't have it on the graph. Or I'm sorry, we don't have it on the poster because there was no correlation whenever we did the stomatal width. But on stomatal length, we can see a slight trend going for where we see higher precipitation, higher stomatal length, lower precipitation, lower stomatal length. We do have a little outlier there that's kind of like throwing off our data, but we also have another May data set that we did not get to analyze because we were having issues with the microscope. So hopefully in that data set, we see some correlation there as well. But pretty much this led us to the conclusion that there is variability among individuals of the same species of little blue stem, which is the grass that we were looking at. And it is related to precipitation in some cases, like stomatal density in the spring and stomatal length in the late summer. Some of the data was a little bit opposite of what we thought there would be. So like for example, stomatal density, we thought with less precipitation, there would be less stomatal density so the plant could retain water, but we see the opposite of that happening. So another theory that we're looking into is maybe the plant has adapted to either not grow or stunt growth whenever there is low precipitation. But that's some more experimentation that we're looking into getting into. But yeah.
Thank you. That's very interesting. For the prairies, what areas did you guys go to collect?
let me zoom in on the map here, and then I could probably pull up a figure where we can show you the exact sites if you're interested, but for the time being, the map of Texas right here shows how it rains more in the east than it does in the west, and the arrows show the direction in which we took samples. Let me pull up my slides from the other presentation and I could show you the exact sites if I think that would be cool to look at. Hold on a second. So yeah, these are the exact sites that we took from. Hi, Sandra. So these are the exact sites that we sampled from, and they're all going across the precipitation gradient.
One more question. So for the so what I'm trying to understand this. So for the precipitation gradient, if it increased, that meant the density decreased. Like if it was more.
That's what
Yeah,
we found. That the density decreased whenever there was more precipitation.
Oh, it's interesting.
Yeah.
How do you think this affects like the overall environment, like with all the rains and like Texas, and now that like the weather has changed more?
so I guess it might affect the growth of the plant because kind of like how Alannah said earlier, we're working on a theory where because how we didn't see a lot of significant results for the late summer samples, and we're thinking it might be because the plants developed a behavioral adaptation instead of a morphological adaptation where it will change its structure. That's changing morphologically, right? But avoidance is when it will just, for example, not grow during times of drought. So, which is why we might not see very significant results in the late summer. As for how it will affect the environment itself, I'm not sure. It's that's a pretty loaded question.
I think keeping towards like more native grasses is one of the issues that we see is growing grasses that are not necessarily native to the region that we're at in Texas. So going towards something that's more understanding how certain grasses are growing in Texas and how they're adapting could lead us to have more native species and just overall agriculture, if that makes sense, or not necessarily agriculture, but in every part of where we're looking at plants growing out in Texas, and kind of like going away from using plants and grasses and whatnot that are not native to the area.
Thank you very much for
Yeah,
the
thank you for
information.
coming.
Have a good afternoon.
You too.
You too.
Can you explain graph number 2 with the
Sure. I'll just give you a rundown of the whole thing, since you didn't get to hear the beginning. So what we're focusing on is how functional traits can show variability within individuals of the same species, and the variability isn't something that we only see among different species. So you probably already know what a functional trait is because of ecology, but just give you a quick definition. Clearly defined characteristics of plants that strongly influence their growth, reproduction, and survival, right? So the functional trait that we focused on were stomata. And the effect, the environmental effect that we chose to study was drought tolerance. So we sampled little blue stem across the precipitation gradient, and you can look at the map here, and it rains more in the east than it does in the west, and the arrow kind of shows the direction that we took the samples in. And I think right as you came in, I was showing a map of where we took the samples from exactly. If you're interested in seeing that again, just let me know. So the method that we used was pretty similar to what we did in our ecology lab, where we took nail polish impressions and we counted the stomata under 400x magnification, and we also took images and we measured stomatal length.
And
But
then-
yeah,
Mhm.
Alannah can go on and explain the results for you.
Yeah, so we have our results here in the middle. The top two graphs are stomatal density, where we didn't see a correlation between the precipitation gradient and stomatal density in the late summer, but we did in the spring. We kind of talked about how that could be due to the growing season kind of like being in the spring, but not really the late summer. And the middle graph, we do see a correlation as far as stomatal density goes. The higher the precipitation, the less stomatal density we saw, which was kind of opposite from what we
Yeah.
were first hypothesizing.
That's what I
Um,
would have thought too. That it would have increased.
That was like really interesting, which is why we're Dr. Tobin and is kind of like moving in a different direction, where we're talking about the morphological adaptation or morphological trait of avoidance, not necessarily growing in low precipitation environments, or like a stunted type of growth, something like that. At the bottom too, we see stomatal length across the precipitation gradient, which is the left to right measurement of the stomata. We do see a slight correlation there with higher precipitation correlating to higher stomatal length. We have a little outlier that's kind of like throwing off our data, but hopefully in our May data set, which we did not get to analyze before the presentation because the microscope was having issues. But hopefully in the May data set, we can see another trend or more correlation that points us into a more defined direction.
So the stomata were not as clumped. They were kind of spread out, but the length of it was larger than what you expected, or were you?
Well, so the length of it, having larger stomata whenever there is more precipitation does kind of make sense.
Because it lets more water come in, right?
Exactly. And having smaller stomata with less precipitation would mean that not as much, that they're not allowing as much water to get out, kind of like preserving their water. So it does make sense. And hopefully the May data set will also show the same trend as well, or something similar. Because we would expect some type of change in traits going along that precipitation gradient.
So maybe you can explain this to me because I'm having problems with this in ecology. The
Sure.
stomata keeps the water inside the plant, or it allows it to come in?
So stomata are bordered by guard cells, right? And I'm just gonna,
Right.
we're gonna go to this picture first. You can see the guard cells bordering the stoma, right?
Mhm.
And a major route for water loss is through these stomata. So
Right.
when stomata are open, the guard cells are open, that allows for uptake of CO2, but that also means that water vapor is being lost from inside the leaf. So there's a trade-off between these two resources. If the guard cell decides to close, which it would usually do maybe in a low light setting or at night, or to preserve water, So if it's closed, that means it's preserving water. There's no water exiting the leaf, but
Mhm.
CO2 can't enter the leaf. And why is CO2 important for photosynthesis? So if
Right.
stoma are closed, photosynthesis is greatly reduced as well.
Okay.
And, earlier, I think Kelly brought up a good question about humidity,
Right.
which, I mean, at that point, you're looking at, if the leaves are in like a humid environment, the stomata would be more inclined to stay more open because it's not going to be losing as much water, if that makes sense. They're already in an environment that has a lot of water if it's really humid. So if it's open, they're not going to be losing as much water.
So it kind of maintains the pressure inside.
Exactly. Yes,
Okay.
exactly.
Yeah, I do that with my plants because I have plants and I usually cluster them together so that they can be humid.
Oh, yeah, smart.
Because if
That's
you
really
if
cool.
you separate them apart, like they
Yeah.
tend to wilt a little more.
Uh-huh.
So that totally makes sense.
That is really interesting. Yeah, I forgot that you're a plant person. That is so cool. Yeah, awesome.
Yeah. Yeah, although I don't know, struggling in that sense, but I'm getting there.
Uh-huh.
I just
That's
thought
really
it was
cool.
interesting that middle graph where it was actually having an inverse relationship.
Yeah, it
So
was really, really surprising. I wouldn't know if surprising was the right word actually, but it was like
you had to
unexpected
think
to say
about
the
it.
least. Yeah, exactly.
Yeah, so when you first see it, you expect things to go a certain way, but then after you think about it more, it makes sense. So
Mhm.
that's pretty cool.
Yeah,
Are you
thank you so much for coming, by the way. I just want to say thank you. Like, I'm so happy you're here.
Well, I mean, it's interesting to me. I was
Yeah.
hoping that you would still be here because I didn't know, like it looks like it's going all day.
Yeah,
So
we're
this
here
is-
until about 2 o'clock. So
yeah. Are you guys going to continue this into the next semester or are you guys done with this?
Mm, I know me personally, I'm supposed to graduate next semester. So
Mhm.
like my course load is a little bit heavier. I would like to, though. It's really interesting. And I'm probably going to be emailing Dr. Tobin if they've collected any more data, because
yeah.
I'd like to see what
Will he continue with
they
this
are.
study? Do you know?
So I will be continuing research with Dr. Tobin next semester. And there are other things that we want to look at, which includes stomatal width. And we also want to look at minimal leaf conductance as well. So this is only like a small part of everything that we want to look at, to be honest. We're definitely going to be expanding more on this and hopefully take it beyond just little blue stem and beyond Texas too.
Okay. Because I'm still undecided, which kind of research I want to do.
Yeah, this is a
Um,
glance at what you can expect. Personally, I'm going to be taking on more outdoor work next semester. If you're interested, you can ask, I can tell you about that later, or you can ask Dr. Tobin about that.
Okay. I'll wait and see how it goes
Yeah.
after this class, and then I might approach him about it.
Yeah. Good luck, girl.
I would
Yeah.
recommend. It's pretty interesting. And Dr. Tobin's like super cool, really nice about everything. So
Yeah, he's super chill.
yeah.
I got an offer from Dr. Morano last semester, and I told her I wanted to take more plant classes, but I don't know yet. So I'm kind of deciding. I like mushrooms, so we'll see. But Tobin is really like I've learned a lot being in his class, and I think I might take plant biology with him next semester. So I think it kind of go with the research.
Hey,
So
Taj, what do you think? You've been quiet this whole time. Let me know if you want us to explain some things to you again, or if you have any questions.
Oh, I was just trying not to interrupt you. Like I left the conversation. I was just trying to finish reading through it. I find this very interesting. So I'm just asking, how long did it take to gather all this data? I don't.
Mm,
So the samples were collected during 2021. I was not working with Dr. Tobin at that point. I started working with him at the last fall semester, basically. And it was after he collected the samples that I and some of my great collaborators, which we see the names of at the top here. We were counting stomata, which also took a few weeks, and then taking all the images and the measurements. It is a very time intensive process because you're just looking through so many samples and slides. Hi, Jose.
Okay. Oh, thank you. Thank you very
Yeah, you're welcome. Let me know if you have any other questions.
much.
Okay, I'll talk to you later about it because I am considering it. And since, it has a lot to do with what I kind of want to do.
Yeah, that's
So-
awesome. Yeah, for sure. I'd love to talk to you about that.
Yeah. Okay. Well, nice to see you.
Okay,
And congratulations on graduating
Thanks
this
for coming.
semester.
Thank you.
Bye, guys.
Bye. Thank you so much for stopping.
No problem.
Hi, Jose. So do we need to explain everything to you, or are you just going to get straight to grilling us?
I would like an explanation. Like, treat me as if I know nothing.
Okay,
Okay.
let's get
Yes.
into it. All right. So, I'll go first and then we'll just see how it goes. Anyways, so the focus of our study was variability of functional traits, not only among different species, but also between individuals of the same species, because most research focuses on variability between different species. So we were looking at functional traits, which are clearly defined characteristics of plants that strongly influence their growth, reproduction, or survival. And the specific functional trait that we were observing were stomata. And the reason why we were looking at stomata was because a major route for water loss is through stoma. And we basically kind of wanted to link it together with like drought tolerance. And the species that we focused on was Zacharyum scoparium, which is also known as little blue stem. The samples were collected during 2 time periods. So spring of 2021 and late summer of 2021. And they were collected along the precipitation gradient of Texas, which if we, if we zoom in onto the map here, we can see that there is a pretty dramatic gradient. It rains way more in the east than it does in the west, and the arrow shows the general direction in which we sampled. We took nail polish leaf impressions and we observed them under the microscope at 400x magnification, and we counted stomata, and we also took images of the stomata and measured their length as well. I think that covers my part, Alannah.
Okay, and then we have the results in the center here. So the first 2 graphs are going to be our first stomatal density across the precipitation gradient. Stomatal density as in how many stomata are in a specific area on the leaf. For our late summer results, we're not showing any correlation between precipitation and stomatal density, but we do see a correlation in the spring. As precipitation increases, we see a decrease in stomatal density. And then for the third graph on the poster, we see stomatal length across the precipitation gradient. With stomatal length, we're talking about the left to right measurement of the stomata. We do see a slight trend on this graph of as far as the increase in precipitation correlating to the increase in stomatal length. We have a little outlier there that throws off our data, but hopefully with the May data set coming in shortly, we'll kind of be able to evaluate that trend a little bit more. But pretty much we're able to see that it's evident that there is variability among individuals of the same species of little blue stem over the precipitation gradient in Texas, because we can see the correlation in the stomatal density graph and then the trend, the kind of going towards the trend and the stomatal length graph. And even though the results are a little bit opposite of what we're expecting, especially in stomatal density. So stomatal density, we would think less precipitation, lower the density, but we're seeing the exact opposite. So we're thinking that maybe the species has developed a morphological trait of avoidance to avoid growing in the low precipitation area of that precipitation gradient. But yeah.
Okay, so I took some notes and I have some questions for y'all. So just to start off, so you mentioned, for instance, that you collected samples during both the spring and the summer. So do you think this has an effect at all as far as your results?
Yeah, for sure. Like if you look at the graphs, we can see that spring is the only one with significant with real significance, like statistically. While summer, we didn't get any relationship for stomatal density and precipitation, but for stomatal length, we did see a little bit of a correlation, but it still wasn't statistically significant. So it's because of this that we are, like Alannah said, theorizing that little blue stem is instead adapting to have avoidance behavior instead of morphological changes according to drought. And that probably did affect the results that we were seeing. Also, hi Razad, I'm so happy to see you here. If you have any questions, let us know at any point.
Okay, thank you. So now I have a follow-up question. From the methods that you did use, did you consider any limitations in them?
I think limitations could come from the quality of the impressions of the methods. The quality of the impressions of the nail polish impressions to be able to look the stomata. I know that some of my data, I ended up having to just not do it because I couldn't get accurate length measurements or a quality image to get an accurate length measurement. So that could definitely be a limiting factor if there was more. And then too, like whenever I was taking the images, there would be some parts of it wasn't really consistent on where exactly I could take a picture and get usable stomatal images to be able to take the links. So if I'm trying to stay in the middle of, and not the exact middle, but like you have the mid vein and then you have the right side of the leaf, the left side of the leaf. If I'm trying to stay within the middle of that area, but there's no usable images that I can take and I'm going towards the end or closer to the mid vein, then that would kind of limit our data as well.
Thank you. And I have another question. So when you mentioned that you had a slight increase in your stomata length. So currently for right now, what all can you say about that?
The slight increase in stomatal length, we can probably attribute to larger stomata in general whenever there is an increase in precipitation. So if we have larger stomata, that means, well, let's look at it on the opposite side. If we have smaller stomata with less precipitation, that would correlate to less water loss because the pore wouldn't be, or the opening wouldn't be as big. If it's if with less precipitation, the opening wouldn't be as big, so there would be less water loss, or at least that's what we would think looking at this trend line. But I would say we can't really conclude anything from it until we get the other data set as well.
Gotcha. And I have one final question. So when you went to collect a little blue stem, for instance, did you have to contact like local botanists in the area to verify that it was the correct species? Or what all processes did you do to verify that it was the correct species?
So Dr. Tobin was the one who collected the samples, and this was before I started working with him and before Alannah started working with him as well. But from what he told us, little blue stem is pretty distinctive, and I'm going to zoom in on the poster here. That's it growing in the middle right now in the picture, and you can see that its color is pretty different than everything else that's growing around it. So I think that in a field of grasses, it would be pretty easy to identify. And another precaution that we took to ensure that we had the correct species was that when we were counting stomata, we made sure to check the upper surface before we started counting the stomata on the bottom surface, because little blue stem should not have any stomata on the upper surface. They're only on the lower surface. And there were a couple of instances, I could say maybe there was at least 1 to 2 slides where we could not count the stomata for that because there were stomata shown on the upper surface of the upper leaf impression, right? So that just probably meant we had the wrong species. And of course, because of that, we couldn't include it in the data set.
I did talk to Dr. Tobin too, and he did contact local like botanists in the area of where he was sampling, mainly because one, to make sure that they could have permission to be able to like go into certain areas and actually take samples. But he said that he was working with, or not working with, but like pretty much talking with other local botanists within the area so they could let him know what areas had like higher concentrations of little blue stem. Like for instance, some of it was like on private property and you can't really go in there without getting the permission from the owner of the private property. So he said that he was like going along the freeway, like, and for the most part going along the freeway and just getting out of his car to like sample because it's Texas and he didn't like want to get shot. So
Oh my God.
but yeah, he did say that he talked with local botanists to at least like get a good area where there would be a lot of public accessible little blue stem.
Well, that actually answered all my questions. Thank you for your presentation.
Great
Wow.
questions. I liked how specific they were.
Thank you.
Yeah.
I do try my best.
Yeah, no, I really appreciate it. Have you been able to visit other posters?
Yeah, I grilled other students so far.
Okay,
So,
that's nice. Making
yeah.
everybody squirm in their seats.
Oh, well, it's more like I'm trying to show that I'm interested. But
Uh-huh.
yeah,
Yeah, that's great. I'm glad you're having fun.
thanks.
Yeah.
Oh gosh. I'll see y'all next week, but definitely it
Okay.
was a nice presentation.
Yeah, thank
Thank
you.
you.
See ya.
Bye. Oh, gotta love Jose.
I was like, I want to show that I'm interested. Do you? Do you?
Or are
though?
you trying to make people uncomfortable? No, he's really kind. I'm glad he was able to visit. Dum-de-dum-de-dum. I need to pay attention to the chat more often.
I know I didn't see that that last message.
Yeah, I just saw it right now.
You said Hatoon calling
Yeah.
you.
How's your week been so far? Oh,
Hello.
uh, hi, Ragad, how are you?
Hey, I just got your text. I was in another poster, but I'm
Were
here
you?
now.
Yeah, okay, I'm
So,
glad you were able to. I was like, oh my God, I need to text her. So I'm glad you're able
yes,
to
and
come
I saw
here.
your presentation in the morning.
You
You
did?
were
You
awesome.
did?
Yes,
Oh, thank
yes.
you. Oh my
It
goodness. I
was
didn't even know
so
you were
good.
here. Okay,
Yep,
thank you
I was
so
there.
much.
I was there. You did very
Yeah.
good.
So you
You were
probably already know
amazing.
everything then. If you have any questions, then let us know. Or any
I do
thoughts
not
or
have
whatever.
any. Actually, let me ask you a question. How
Mhm.
did you start? Like, how was the process of starting? Maybe it's not related to research, but like the research in general.
So and
Like, how did you end up with this poster, you know?
sure, yeah. So the poster was, in collaboration with me and Alannah. I just slapped some stuff on there and Alannah rearranged it really nicely and she added her own touch to the poster and made it look a lot nicer than how I made it.
Yep. Hey,
So
Alannah.
Hi.
yeah. So I guess when you're making a poster, because everything is going to be, so normally this would be a huge, right? Like you would be able to step up to it and see everything pretty.
Yep.
But
Yep.
because it's online, we try to take into consideration the fact that we couldn't put too much text, otherwise it's going to be hard for people to read the poster.
Yeah.
So we try to make it as many diagrams as possible and give more visual guides than more text to read, basically.
Okay, so what's your research about? What was it?
So we were studying functional traits in plants, and the specific trait we focused on were stomata, and the environmental variable that we were looking at was precipitation. So we were trying to see the variability of functional traits within a species called Schizachyrium scoparium, which is also known as little blue stem. We're trying to see the variability of this functional trait across a precipitation gradient in Texas. And here's like the map up here, which shows the precipitation gradient. And you can see that it rains pretty heavily in the east and not as much in the west. The arrow shows like this direction that we sampled in. And we made leaf impressions using clear nail polish. We counted stomata under the microscope. We also took images of the stomata and used those to measure the lengths. Let's see. Yeah, I guess Alannah can explain the results now.
Yeah, so we have our results here in the center. The first two are our stomatal density for late summer and spring. We would have had a spring for stomatal length here at the bottom, but we had issues with our microscope and we were not able to get that data in before the presentation. But pretty much for stomatal density, we're looking at how many stomata in a given area on the leaf. For late summer, we didn't see a correlation with that for precipitation, but we did see a correlation in spring. In spring, we saw that the more precipitation or the increase in precipitation correlated to less stomatal density.
Wow.
Which was, yeah, so it's really like surprising because you would think that you would think that in a drier environment or less precipitation that there would be less stomatal density to so that way there's not as much water loss in the plant. But we saw the exact opposite. As far as stomatal length goes, we saw like a trend, as you can see in that bottom graph. But we have a little outlier that kind of like threw it off. But we saw a trend where the more increase in precipitation, the larger the stomatal length. And stomatal length would be like the measurement from left to right of the stomata. Hopefully in the May data set, we see something that kind of goes along the same way so that way we can make some like definitive conclusions.
So you're still working on it?
Well, sort of. So, so we got all of like the images down and everything, but we're waiting on one of our partners to analyze the data.
Yeah.
But our microscope went down and we have not been able to get it back up. So we had to use a different microscope to finish off the data set.
That's actually pretty cool. So
Yeah, we're also going to be looking at other things. Like Alannah
Mhm.
said, also the other sample sets for spring, and we're looking at stomatal width, and we're going to be looking at minimal leaf conductance as well. Or yeah, minimal leaf
that's
conductance.
for next semester or like still like right now?
We are coming to the end of the semester, so
Yeah.
I think anything else that we can fit during the semester, maybe just analyzing some of the data, maybe we can get that in there. But as for other factors like the leaf conductance, I think that might have to wait until next semester.
Oh, well, I'm gonna be with you next semester, so.
Yeah, I hope so.
So, hi. Yeah, I'm gonna do the paperwork hopefully today. I don't know.
Really? Okay, great.
Yes.
Congrats.
Yes,
Awesome.
thank you. We'll see.
Yeah.
It's really nice meeting you all. It's really nice meeting Alannah, but it's nice to see Hatoon again.
Yeah, it's great to hear from you too. I'm so happy that you're able to.
I know whenever I saw the text, I was like, oh, please don't be late. Like I didn't want to be late. I immediately like jumped to your poster.
Yeah.
I was like,
Yeah, thank you so much.
You're welcome. Well, I'll see you when I see you, okay?
Okay,
Thanks for
inshallah,
coming.
sure. Bye.
Bye. Thank you, Alannah. Bye.
Bye.
Hi, Kishan.
Wow, what a beautiful poster.
Thank you. Thank you so much. Alannah, take a bow. Every time somebody compliments a post, I'm like, Alannah.
It's
Yeah,
magnificent.
we're like, it needs color. It needs color. It looks
Yeah.
boring. We found
No,
a
it-
couple of colored images because even then, we like the microscope images. I was like, crap, these are all like black and white too.
Yeah. No, I like it. It looks really good. Magnificent.
Thank you. Thank
Who
you.
is
What's up?
it's exquisite.
Yeah. Oh my goodness. Like more. What other adjectives do you have stored up your sleeve?
Phenomenal.
Oh my god. Oh, that was too strong.
I'm flabbergasted.
So do you
Did I
have
get
any
a lot?
what
Huh?
now?
Oh, did I get a lot of questions?
Yeah, we're getting some really good questions so far. Jose came and asked some pretty nice ones. Hi, Bianca.
Hello,
How are you doing?
I'm good. How are you?
I'm great. Thank you for asking. Do you want us to give you a rundown on our presentation?
Yes.
All right, I'll go ahead and start then. So we're looking at functional traits and how they can show variety among individuals of the same species and not necessarily between different species. A functional trait is a clearly defined characteristics of a plant that influence their growth, reproduction, and survival. And the trait that we were looking at was stomatal, not stomato, just stomato. We were looking at stomato. And we were studying how drought tolerance can affect the factors of this trait. So the species that we chose to focus on was Schizachyrium scoparium, which is also known as little blue stem. And we sampled this grass all along the precipitation gradient, which you can see in the map here. And it rains way more in the east than it does in the west. And you can see the arrow, which points in the direction that we kind of took the samples across. We measured stomatal density by making impressions using clear nail polish, and we counted stomata under a microscope at 400x magnification. And we also took images of the leaf impressions and used those to measure the length of the stomata using Fiji software.
All right, and then we've got our results in the middle. So the first two graphs are going to be for stomatal density for late summer and spring. Stomatal density referring to the number of stomata in a given area on the leaf. For late summer, we didn't see a correlation between precipitation and stomatal density, but we did see one for spring where the increase in precipitation correlated to a decrease in stomatal density. Our third graph there, we see stomatal length, which if we look at this bottom left image, those are the microscope images that we were taking of the stomata, and we're measuring from left to right of the guard cells there. So we do see like a slight trend line for the stomatal length in the late summer. We have a little outlier there that's kind of throwing off our data, but for the most part, we see that an increase in precipitation is correlating to an increase in stomatal length. And we don't have our May data set in because we're having some issues with our microscope. So we just recently got those images in this week, and we still haven't analyzed like the measurements for them. But hopefully whenever that data set comes in, we also can see some type of trend line going in the same way. But this information is kind of surprising because as far as stomatal density goes, we were thinking that a decrease in precipitation would correlate to a decrease in stomatal density. Because when we talk about stomata, we're talking about like water leaving through that little pore, that little opening. So we would think that there would be less of those whenever there's less precipitation, but we saw the exact opposite of that. So there are some other traits that we're going to be thinking about looking into, some different theories as far as the plant taking on a trait of like avoidance and not necessarily growing whenever there's less precipitation, rather than modifying their stomata for to be able to tolerate drought, if that makes sense. But yeah.
Let us know if you guys have any questions or you just want to talk about some stuff.
I had a question actually. I was wondering why this species, the Schizocycrum scoparium, was studied like particularly.
So it's a pretty common prairie grass, and it's native to eastern Texas. And I think I need to emphasize how it wasn't necessarily the species of grass that we were focusing on, but more like how functional traits can vary within a species and how drought tolerance can also like have its effect, right? So the reason why we chose this species was because, like I said, it's a dominant component of a tall grass prairie, and it's found all along the precipitation gradient of Texas. So as Dr. Tobin was going along and collecting samples on this arrow here, he could find little blue stem all over it, and that just made it a very practical choice for sampling as well.
Okay.
I had a question as well. So you had mentioned about the samples that you collected, that it was across Texas from west to east, right? Or from east to west.
Yeah.
So how were they collected?
Alannah, do you want to take that question? I thought we were
Sure.
trading off questions. I was waiting.
When you're collecting little blue stem, pretty much you have, can you zoom in on the picture there?
The left
Of little
one?
blue stem? Yes, please.
Oh, this one?
Yeah. So little blue stem, you're gonna see like the leaves kind of like growing straight up, like you see in that picture there. So in order to collect them, you're looking for the leaves like kind of towards the middle that aren't necessarily that aren't like new grown leaves, but they're not old leaves and they're not too dried out by the sun. You're kind of looking for like that perfect like mixture of like a good sample of the best of that plant. So that way you can get good images and good data. But pretty much you go towards the base of that leaf and then you just clip it and you take a few samples from each plant, save them, and then end up being able to collect some data from them.
Okay, thank you for answering my question.
Yeah, and if you guys have any other thoughts, then let us know.
I have a question for the figure in the middle.
All right, the stomatal density.
Yeah, so I know Alana was saying like it didn't produce like the data y'all were like hoping for, because the trend is showing that, is that showing like with increased precipitation the density decreases?
Yeah. So the normal thought process would have been as precipitation increases, we would have a higher stomatal density because the plant wouldn't have to reduce water loss by closing the guard cells as much as it would have to if it had less precipitation.
Yeah.
So it was just like kind of like one of those surprising results.
That's interesting.
Mhm.
Very
Am
interesting.
I right?
Yeah, I think especially if we look at it on the opposite end too, like when we're talking about drought, I mean, we're not necessarily looking for like the increase of stomata, but we're looking for like the decrease of stomata when we're looking at less precipitation. So to see that actually increased, I mean, I would have even been more, I would have been less surprised to see that there was no correlation at all than to see that there was an increase with less precipitation.
Yeah, that's true. At least there's some kind of trend, right?
Whatever that says.
Yeah.
Yeah, it was a really good post for you guys. You did
Thanks,
awesome.
man. Thank you.
All right, I'll see you guys next week.
All right, see you.
Next week.
All right, bye.
Bye. It feels like time moved so fast.
Really? I'm looking and I'm like, there's still 10 minutes.
I'm surprised. I'm like, oh my God, we only have 10 minutes left.
I think for the next set of poster presentations, though, it's shorter, huh? Like only an
Yeah,
hour.
it's only an hour. I have no idea why they put it like that, but I don't know. It was kind of weird.
Matthew. Yeah, because it looks like in the second presentations, there's even more posters too. So I don't know why I would have less time.
Yeah, that is weird. I don't know. I feel like they're about the same amount of posters actually. Maybe one or two more.
Really? Maybe I just miscounted. 4, 1, 2, 3, 4.
I'm just kind of eyeballing it to be honest. I'm not really mathing it.
Oh, I think I think it's like 5 posters more on the second poster session.
Oh yeah, and there's like one graduate level project. That's interesting.
Where is that at?
It's like all the way at the bottom right for the second page of like the poster sessions.
Oh, okay. Oh, is it? When is Earth Day?
I think that's today.
Really?
Yeah, there was like a, you know how Google has its like little art thing.
Oh, yeah, I see it.
Yeah, it's pretty cool. There was one image that I recognized just because of Dr. Tobin's ecology class. It's not the same exact thing he was talking about in class, but it's about glacier retreats. And they had an image of like a glacier, like how it looked before and after a retreat. And I was like, I know that. But yeah, felt pretty smart for like 2 minutes.
I didn't take them. Well, I never took ecology. I took them for plant biology.
Mhm.
And
Yeah, I'm
I
taking that next semester.
think I think I got the question wrong whenever it was like, I remember one of his questions was like the difference between ecology and something else or whatever. And I think I got it wrong because I was like, I don't know.
Oh, let me guess, because he asked us a similar question too, like the difference between ecology and like an environmental activist or something like that.
I think it was like, crap, what was the question? I want to say it was something like ecology and like, or ecology and like ecosystems or something like that. Something that was like really similar. And I was like, I
Oh,
don't know. I
okay.
still don't know the
Yeah. That's that's so fun. Well, I'll cut that out of the recording for you. Actually, I can't because I don't know how to, but you know, maybe it's the thought that counts, right?
If he goes back and watches it,
Hi.
sorry. What are you doing your degree in?
Biology, just
Oh,
pre-med.
okay,
Yeah. What about you? We never really talked about
I
like
know.
simple stuff.
I'm doing biotechnology.
That's cool.
No plans for afterwards, just kind of like going where the wind takes me.
Really.
Yeah.
Are you not nervous about that though?
No.
No? Okay, that's pretty cool. I admire that.
Yeah, so whenever I talked to Dr. Tobin, he was like, if you're wanting to like, because I was like, well, I think I just want to work in a lab or something. He was like, you say that until you get in there and it's so boring, blah, blah, blah. He was like, you should like get a lab job first and see how you like it. So then I got hired at Memorial Hermann to work
Cool.
in the cytology department,
Uh-huh.
which is like No, like nobody ever knows what that is, but do you know what histology is?
Yeah, it's like the tissues.
Yeah, so cytology is like taking cells like from bodily fluids or from liquids, and then evaluating it from there.
So that's like where all the blood samples go when they take your blood tests, I mean.
Not blood samples, but we get like abdominal fluid, thyroid fluid, cyst fluid, lots of different fluid, cerebrospinal fluid,
Oh, wow.
like a whole bunch of fluids that I was like, oh, I didn't even know that these were actual fluids.
That's pretty cool.
Yeah, but they send us all of that. So like I'm working in a lab every day now, and I don't know, I'm having a good time with it. And then I was talking to Dr. Visbal, and Dr. Visbal was like, oh, I can put you in contact with like some of her colleagues that work in like histology research labs. So I was like, well, that seems like it could be cool too. I don't know. I kind of just want to see like what's out there, like what cool labs I can get into, because I do like biology and the biological sciences. And my first pick, well, this is no surprise. I told Dr. Tobin this, so if he watches this, I think my first pick was like I wanted to work with Dr. King with like the flies, like doing like the genetic type stuff.
Yeah.
But I kind of like applied really late, so she wasn't available, but she was like, oh, I think Dr. Tobin is looking for people, and I was like, okay. But yeah, I don't know, working in some type of like lab like that would be cool, or like some type of like biotech startup company would be super cool. And then Dr. Tobin was like, oh, don't go on to like graduate school or anything unless somebody's paying for it. He was like, you don't ever have to pay for that, blah blah blah. So I was like, that's good advice.
Yeah, exactly.
So I'm like,
that's true.
Could be in the future, graduate school could be in the future if the opportunity comes along.
All right, yeah, that's pretty awesome. I mean, and it's not like you have to go into graduate school immediately either. I think the thing is that you can take some time off and just use that to try to get more experience.
Yeah, make more connections and get more experience. That's kind of like what I'm looking for.
Yeah, it's pretty awesome. I love that.
And you said you're doing your pre-med?
Yeah.
That's exciting.
Yeah. Yeah. I'm not applying this year because I'm a junior, right? So I will be taking a gap year, so I'll be applying May of next year.
Okay.
Just because like with the pandemic and everything, it was super hard to get like as much experience as I would have wanted, like with volunteering and shadowing and that kind of stuff.
Yeah.
So, you know, it's still something that's in the process of like being made or happening, you know.
That sounds really cool though. What do you want to do in med school?
So I'm really thinking about specializing in dermatology.
Oh, okay.
I think that the skin is really cool, and especially nowadays there's a huge demand for dermatologists because a lot of people, they're becoming way more conscious about like how to keep your skin safe and that kind of stuff, like sunscreen, and like skincare is just so big right now.
Yeah.
And then like of course dermatology is like a little deeper than just like skincare, but I feel like it's a really great field to get into. I'm really excited about it. It's something that I really want to look into. And I'm kind of thinking like pediatrics. Maybe it's like, I don't know. I'm kind of unsure on that one just because apparently I'm really good with kids, but I don't know.
Aww.
Yeah. I say apparently because I feel like kids hate me, but I don't know. Like there's supposedly two different views on that.
Well, you're like super hardworking and like super diligent, so I know that you're gonna do something great and something good.
Oh, you're so sweet. Thank you. Yeah, thank you so much.
Like I know med school takes like serious like determination, or even like applying to get into med school takes like serious determination. Like
it is, yeah.
I personally like couldn't take that route because I'm like, ooh, I don't know if I'm that dedicated. Like I know grad school is like kind of different, but I'm like, I think there's like differences each route that you go, and med school is just a little bit Ooh, that's like a
Just a little bit too much of a reach.
it's a little bit too much of a read. You got to be
Yeah.
like real diligent and disciplined and everything.
Uh-huh.
But you seem like you're gonna do like really awesome.
Thank you so much. Yeah, I really appreciate that confidence just because I need it. Yeah. They say that like the imposter syndrome, I don't know if you feel the same way I do, but imposter syndrome never goes away, apparently.
Dude, I can't, sorry, I didn't mean to cuss out, like, because it's being recorded, but like,
Yeah.
it's ridiculous. It's ridiculous. Like, literally, like in my family and like my friends, they're like, oh, you're the smartest person I know. Like, you're so into your studies, you're this, you're that. And I'm like, dude, I feel like I'm just like,
I'm
I
just
don't know what I'm
vibing.
doing here. I don't know what's going on.
Yeah. Oh yeah.
I feel like I'm here, but like, am I supposed to be here? I don't know.
It's like, do I really know what I'm saying and doing? I don't
100%.
know.
Like, I don't know. I'm kind of just winging it, and people
Mm.
seem to think that I'm doing the right thing. So
Yeah, personally, I can say that I do like your attitude about everything, and I also think that you're gonna go on to do pretty great stuff. I love how flexible you are. I think that is such a great skill to have.
oh, thanks.
Yeah. So, you're gonna do awesome.
You know, my manager, I was like, she's like, oh, you're graduating with in biotechnology. That's a pretty broad subject. And in my head, I was like, I didn't think it was whenever I like was choosing it as my major. I was thinking like, oh, it's a little bit more specialized and we can,
Yeah.
and she was like, no, it's pretty broad. So, and I'm like, well, I guess keeping it broad is key,
Yeah, that's especially good for STEM majors because a lot of STEM majors, like my biology degree, it would be pretty hard for me to get into work if I didn't want to go into med school or graduate school. But biotechnology is also something that I think is like really expanding.
Yeah, it's
A
like
great field
super
to get into.
expanding a lot right now. So I'm really excited to try, like my goal, like fingers crossed or whatever, is like I'd be able to get into like a really cool like startup biotech company and kind of grow with them from there. Like that's like the
Yeah,
ultimate goal.
that would be
But
awesome.
we'll see. We'll see what happens.
Yeah. Best of luck, girl. You
Best
got
of luck to you too.
Mhm. I think our poster session is done. Like,
It is done. It is over.
oh, we got through it.
Oof.
Okay, I think I'm gonna, let's see. I need to stop recording.